Hello, my name is Neil Spooner. I'm the chair of the Patient-Centric Sampling Interest Group, and I'd like to welcome you to this month's free PCSIG webinar. This month, I'm really excited to welcome back Christophe Stove, professor at Ghent University in Belgium. Christophe is a long-time friend of myself and Patient-Centric Sampling Interest Group, and he last spoke to us in October 2023. I can't believe so much time has gone by since that occasion. So I'm really looking forward to the updated information from Christophe. His talk is entitled Microsampling from Promise to Implementation. Before we start, it's just an important time. We wouldn't be able to put on these webinars without the generosity of our sponsors. So we've just started a new sponsorship year, and these are all our current sponsors. If anyone's interested in sponsoring the organization, please just contact me at neil@pcsig.org. There will be, at the end, an opportunity for you to submit your questions. Please do them through the chat function. And please note the webinar is also being recorded and will be available free in a few days through our YouTube channel. No further ado, I'd like to hand it over to Christophe, and welcome again to PCSIG, Christophe.
Okay, thank you. I'll see if I can get the pointer to work. Hold on a second. It says, you're still using it, Neil? I believe.
I don't know how to stop using the pointer because I didn't know I was using it.
Christophe, if you just use your up or down arrows, that'll give you control of the slides then.
Okay. Yes. Okay, good. So, welcome everyone, and thanks for having me again. I'll be talking on microsampling from promise to implementation. We all know that microsampling really took off some 10, 15 years ago. And aim of this presentation is actually to see where we are at this point. Have we really come to implementation? And if not, what's still needed? And so actually, while preparing this presentation, I thought, oh, probably AI can do the thing. And so I actually prompted to Bing Image Creator generate a picture representing patient-centric sampling. And so this is actually the result that I got. Patient-centric sampling. Okay, the sampling is in the center, but yeah, I still see here someone involved. I see a tube here. So I said, no, that's not what I want. Do better. And next was something like this, and then I got, so it's already better. It's like someone in the home environment and could be, but still there's a tube of blood here. So I'm wondering how this individual collected that tube of blood. So we're not there yet. If we think about, let's say, the comprehension by AI of what patient-centric sampling is about, and probably that will also be the conclusion of today. We're not there yet. But before coming to that final conclusion, obviously, I'll try to guide you through. In a sense, what I typically start with is an overview of what can be detected in a blood microsample, a dry blood microsample, because that's typically what can be collected in a home environment. And in a sense, almost anything. You can look at DNA, mRNA, you can look at trace elements, you can look at proteins, you can look at small molecules. And if we think about the context of patient monitoring from home, and if we're thinking about therapeutic drugs, then here the small molecules are still the mainstay, although there's more and more biologicals. So actually, you could also say that proteins may also gain interest if you consider the follow-up of patients via microsampling at home. Why would someone be interested in microsampling? There's a wide variety of reasons. And in a sense, you could say, actually, because it allows you to do things that otherwise are not or are hardly possible. And then you can think about preclinical studies in which you cannot predict when patients will need to take their medication. Clinical trials, post-marketing studies in which you may wish to evaluate thousands of individuals, and that's not so easy if you consider that when everyone has to come to a clinical center. Epidemiological studies, I'll provide some examples, and we've involved in quite a few epidemiological studies now, and we still learn from those actually. Then an important application, if you think about the monitoring from patients at home, is therapeutic drug monitoring at home, a TDM at home, and also remote sampling, for instance, for toxicology purposes, or if we also think about toxicology purposes, sampling by non-specialized individuals, in which you can easily have supervised sampling by a non-specialized individual, so you don't really need a medically trained person. And if we think about the type of analytes in this context, could be therapeutic drugs, could be illicit and abused substances, vitamins, biomarkers, and yeah, sort of everything, whatever your marker of choice, your analyte of choice could be. In the context of today, I'll primarily talk about therapeutic drugs, biomarkers, and therapeutic drug monitoring at home, because that's primarily the field that we are working in, but also some toxicology I'll touch upon. Good, I'll be brief on this and the advantages and challenges associated with microsampling. In a sense, you're collecting a small volume in a minimally invasive way. It's an easy sampling procedure. In a sense, almost everyone can do it. It can also be quite an economic procedure. If you look at the overall costs of the sampling, I'll get back to that. It's amenable to automation, also sample preparation, simplification, convenient storage and transport, just at ambient temperature is quite often possible, and quite often it has a stabilizing effect. If you think about animal sampling in context of laboratory animals, then it also conforms to the 3R principle, because you can reduce the number of animals, and it's also a refined procedure. So from one single animal, you can have repeated samplings rather than having one terminal bleeding, for instance. Obviously, there's also a backside of the coin. The small volume is an advantage, but at the same time is a disadvantage, because you're confronted with a very limited amount of sample, which quite often imposes the need for analytical instrumentation that has a high sensitivity. And actually, one of the key things that has propelled the field forward was the availability of high-end instrumentation, high-end analytical instrumentation, primarily tandem mass spectrometry, that was capable of detecting very low amounts of analytes in small volumes of blood. But very sensitive instrumentation also typically implies expensive instrumentation. There's also potential capillary venous differences. So if you sample from the arm, you may have a different result than if you sample from the fingertip. There's some interpretative issues, blood versus plasma. Actually, just today, a publication came out in which we tackle that. I'll also touch upon that later in the presentation. You can still have stability issues. Sample quality is important because you don't want to work on a blood smear, but on a high-quality dry blood spot or dry blood sample. It's not always dry blood spots on filter paper. And then there's some specific issues related to dry blood microsamples. You may have recovery issues, hematocrit effect, volume issues, spot inhomogeneity. So there's several things that need to be thought of, that need to be taken care of. If we think about one example that we've been working on in my lab for some time now is microsampling in the context of therapeutic drug monitoring. And you can look at a wide variety of analytes. Just some examples are given over here. Anticonvulsants, for instance, antipsychotics, antidepressants, antibiotics, immunosuppressants. So there's a wide variety of analytes that's possible. Just one example, immunosuppressants. It's finalized work, but not published yet. Actually, what we looked at is we took venous blood samples from individuals and generated venous dry blood spots from that, and compared the results with venously collected whole blood samples, so the conventional strategy. We looked at tacrolimus, which is an immunosuppressant, and also at creatinine. So in the same dry blood sample, we looked at two analytes which were quite different. One is typically measured in blood, the other is typically measured in creatinine, and we actually applied a blood plasma conversion. And actually what I show you here is on the y-axis, is comparative evaluation of dry blood samples with whole blood versus the hematocrit, because we know the hematocrit can have an effect. And actually what we want is that the vast majority of the results is within the light green area, so within 20% of the reference, which is either whole blood or plasma, depending if we look on tacrolimus or creatinine. And we see that if we look at the concentrations that were derived from capillary dry blood spots, that actually the vast majority is within 15%. So 98% of the tacrolimus results and 89% of the creatinine results were within 15% of the whole blood and plasma results. So actually this shows quite a good clinical agreement. We have now also proceeded to home sampling of patients, and actually could demonstrate that also the precision that we get from home samples and that were sent to the lab was actually as good or close to being as good as samples that were collected in the lab. So from the sampling up to the analysis, you actually didn't have an increased imprecision imposed by the home sampling. So showing that it actually really can work. Another example that we apply in my lab is in the toxicology context, in which we look at the alcohol biomarker phosphatidylethanol, briefly PEth. PEth is incorporated in the red blood cell membrane if you drink alcohol. And because we are working on red blood cells, you can also look at blood as a matrix. And actually what we showed before was that the concentration in dry blood spots was the same as the concentration in blood. Again, here we're looking at PEth concentration in liquid blood, here PEth concentration in capillary dry blood spots. And if we plot those, actually we get almost exactly the same results. The additional advantage of working with dry blood microsamples in this context is that we have an improved stability, that this also facilitates storage and transportation. We have worked with labs from abroad that have sent samples to us, and those samples were just sent at room temperature via FedEx, and that didn't pose any issue with respect to, on the one hand, the stability of the samples, but on the other hand, also was a convenient and economic way of getting these samples to us. If you would send samples via a dedicated transport from abroad to our lab, and it would be liquid blood, and then it would be way more expensive. And there would be also some issues related to biosafety. For instance, if there would be samples that are HIV positive or hepatitis virus positive. And this isn't a problem if you work with dry blood microsamples. Actually, in my lab, we sample individuals that have to demonstrate that they're abstinent to get their driving license back. And we apply this procedure routinely by performing a finger prick in those individuals and having those individuals come back every 4 to 5 weeks so that we can really monitor them. Because this biomarker allows to look back in time and allows to make a statement about whether you have been drinking the past month, and if you have been drinking, whether you're a social drinker or an excessive drinker. We're now working on automating this procedure because we get quite some samples, and it would be more convenient if it would be a fully automated procedure, obviously. With respect to this biomarker, there's now a trend towards internationally accepted cutoff values, and there's more and more labs actually that invest in this, and actually more and more labs that also are interested in finger prick sampling because of the convenience. There's not so many procedures, I believe, and that are fully accredited and that start from dry blood microsamples. And actually, the procedure that's running in my lab is fully accredited according to ISO 17025 standards. So we do take part in proficiency testing schemes, and actually it's accredited on 2 different platforms that we have in our lab, and one serves as a backup for the other, because obviously if we run this procedure routinely, a machine always has to be up and running. Sample prep is quite straightforward. So you have an absorbent tip. In this case, it's a Mitra device that collects only 10 microliter of blood in a volumetric way. We have a simple liquid extraction, and then we move to the LC-MS. Actually, we generated a dedicated algorithm in order to monitor patients' alcohol drinking behavior via setting up a population study that looked at the kinetics of the disappearance of this alcohol biomarker. In a sense, if you would have been drinking alcohol the past month and you would not be drinking the upcoming month, then we can predict after, or then we actually can make statements after 4 weeks whether actually you have been abstinent, yes or no. And that's not so obvious to make a big study on that, to set up such an algorithm. And that's why some 6 years ago already, we teamed up with an initiative in Belgium that's going to Tournée Mineral. It's also known as Go Sober or Dry July, depending where you're living. And actually, what we did was we recruited hundreds of individuals in a short period of time that were drinking alcohol normally. And then those individuals abstained voluntarily because they took part in this initiative, which in Belgium was in February 2019. And what we actually did was only made possible because of the availability of microsampling. So the individuals didn't come to our lab, but actually we sent sampling kits to all these close to 800 individuals and asked them to sample early February, mid-February, and end of February. And actually, like that, we collected thousands of samples. So those samples were just sent back to the lab. And that allowed us to generate a big database with a lot of data, with initial values of this alcohol biomarker, and then also the decline of this alcohol biomarker over time. And this definitely wouldn't have been possible in a conventional way, because we definitely didn't have the capacity to have 800 individuals come 3 times in 1 month to our lab and sample out all those individuals. It was made possible via home sampling by these individuals and then sending the samples just to our lab. So this is actually what we could generate. We generated some kind of algorithm. And so if you have the initial value normalized as being 1, then we can see here you see the data sets mid-February, end of February. And actually we use this now also even for judicial purposes. So if someone needs to demonstrate that they have not been drinking the past month, we can use this algorithm. And if someone has a starting value over here where the pointer is, and after 4 months ends up of being over here where the pointer is now, then we see that this is above actually the prediction interval, and we can say you have not been you haven't been abstinent during the past 4 weeks. If someone is within this region, then we can say, okay, this is compatible with having been abstinent. So this is a nice example of how we could generate data in a way that otherwise definitely wouldn't have been possible. So you could say, okay, I've already shown several successful examples, both in TDM and toxicology. You could argue, okay, been there, done that. Microsampling is ready for implementation. However, I would say that's not really the case. About 10, 15 years ago, it was hyped a bit, and it could have been some over-promising. And if we look at the uptake by the clinical community, we can definitely say that it's slow, actually too slow, you could say. And if we look in implementation in routine care, then we must admit that actually this implementation is quite rare. In the Netherlands, there's a few labs that really apply it in routine care. We apply it also in routine toxicology cases, but we are the exception rather than the rule, I would say. So how does this come? How can we accelerate the implementation? How can we move from this to here? Actually, there's several steps that need to be taken, in my opinion. First of all, good science and good studies are needed. And those studies should produce solid data. Because provided instrumentation is sensitive enough, I always say that anyone can measure anything in a dry blood microsample. If you have a good system and a sensitive system, you will get a result. The question is the measurement actually right? And is the methodology robust? What we typically do is we try, for instance, if we can get the same result from a 3-hour-old dry blood microsample versus a 3-week old dry blood microsample. And I know that for many methodologies, that's not the case. And that means that depending, for instance, on the age of the sample, the results that you will get may be different. And that's obviously not what you want in a clinical context, because sometimes a sample may have taken 1 day to get from the patient to the lab, or sometimes it may have taken 4 days. And the results should be the same, obviously. We don't want a different result depending on how long a sample has been in the mail. And this has nothing to do with stability. It has primarily to do with extractability, how good you can get the analyte out of the dried sample. So that's why a couple of years ago, we, together with other colleagues from the International Association for Therapeutic Drug Monitoring and Clinical Toxicology, in brief, IATD-MCT, as we set up a guideline on the development and validation of dry blood spot-based, or in general, dry blood-based methods for therapeutic drug monitoring. But actually, the same guideline, you could say, can also be applied for a variety of purposes. In a sense, we say that you should not just try to make a comparison between a capillary dry blood sample and a plasma or serum sample. And sometimes people do this and say, okay, we see that results correlate. But correlation does not mean that you can derive actually the plasma or serum concentration from the capillary dry blood concentration. And I would be surprised if you would not find a correlation between a blood sample and a serum concentration. So that's why more comparisons need to be done. And I won't go into too much detail, but for instance, you need to make sure that if you look at a venous blood sample, a liquid sample versus a dried sample, that you get the same result. In a sense, if you would look at 10 microliter of liquid blood, or if you would look at 10 microliter of dried blood, you should get the same result. So this has to do with the robustness of the extraction. Like I already mentioned, you can have differences between blood and plasma. Several reference concentrations, certainly if you think about therapeutic drugs, are expressed in plasma or serum concentrations rather than blood concentrations. And so this makes the interpretation of a blood concentration more complicated. I'll get back to that later. And also, there can be a difference between a capillary capillary concentration and a venous concentration. In other words, if you determine something from a fingertip or from your arm, you may end up with a different concentration just because of physiological differences. That's something that you can't help, but that you should be aware of. And only if you have done all these comparisons, then you can really make a real valid statement about whether or not your capillary dry blood concentration can really trustworthily can be converted in a plasma or serum concentration. I won't go too much into detail about the hematocrit, but actually dry blood microsamples and have been plagued, I think I can use that word, quite a bit with the hematocrit issue. And in a sense comes down to the fact if you use conventional dry blood spots generated on filter paper, and you apply the same volume of blood with a different hematocrit, then blood with a higher hematocrit will generate a smaller blood spot than blood with a lower hematocrit. Because blood you can be looked at as a liquid. Here, this is a more viscous liquid with a higher hematocrit. This is a less viscous, a more watery liquid, you could say, with a low hematocrit. So if the blood spreads more and you make a sub-punch, which is typically done if you look at partial spot analysis in conventional dry blood spots, then this implies that you have less analyte here and more analyte here. And this under or overestimation that is the consequence can be quite similar, can be quite significant, can be up to 30 or 40% that you're actually off what the actual concentration should be, just solely because of the spreading of blood on filter paper. So that's why different approaches have been put forward. Just listing a few ones here. There's other and more recent ones as well that are there. So there's a variety of devices that allow volumetric sampling. And actually, this comes down to the principle that those devices allow to collect a fixed volume of blood. So with the idea that if you collect 10 microliter and you evaluate this 10 microliter of dry blood, you should get then really the result of 10 microliter. An additional advantage of many of these devices is that they also really aim at facilitating the sampling in a home sampling context. So that's one way how to cope with the hematocrit issue. Another way is, there's here just a few publications that list this volumetric sampling. Another way is try to derive the hematocrit from a dry blood microsample via an approach that you could call hematocrit prediction, or a way in which you can actually derive the hematocrit from a dried blood, which isn't so easy. But again, there's several approaches available. Several are listed over here. So if you want to know more, you can look into that. I'll go a bit more into detail about one of the approaches that we developed. This is one example. This was the lab setup, and actually here you're shining a light bundle on the sample, which is over here, and you're looking at the reflectance of the lights. And to make a long story short, eventually this was incorporated in the system, and the system just scans a dry blood spot. It scans the part where a dry blood spot is present, also scans just the filter paper without a dry blood spot, and from that you can derive the hematocrit without actually consuming part of the dry blood microsample. There are several approaches that are available also for instance, volumetric absorptive microsampling tips if you're not using cards, but again, I won't go into too much detail on that. Just one example to show that the hematocrit that is derived from that can be used to correct the results is given here for tacrolimus. If we look at the difference between a dry blood spot result and the whole blood result for tacrolimus, and that was determined in regular filter paper samples using a device shown over here, then we can see a hematocrit-dependent difference, which is obviously not what we want, because depending on the patient's hematocrit, we will get a result which is either too high or which is either too low, which is not good. There were no ways to optimize the extraction, because that could, in theory, be a way, but we were limited in what we could do with the extraction. So we had to try to find a correction algorithm. If we know the hematocrits of these blood that was used to generate these dry blood spots, we could correct this, but this was a hematocrit that was derived from the blood tube. And obviously, if a patient samples himself at home, you don't have that hematocrit. So that's why we applied one methodology using UV-Vis spectroscopy and an algorithm, and we were able to correct this hematocrit effect. You can also look at the near-infrared spectrum of the dry blood spots. And again, this allows you to correct for this hematocrit effect. So you get a result in which the vast majority of the results is within 20% of the target. This doesn't only hold true for tacrolimus, but also holds true for other immunosuppressants shown here for sirolimus, everolimus, and cyclosporin A, so which was published just 2 years ago. So getting back to this complicated comparison, in which I already referred to the fact that venous blood sample and a plasma serum sample, you may not always find it easy to convert a blood concentration to a plasma concentration. And just today or yesterday, a publication came out again from the ITMCT that actually really explains in a step-by-step way how to convert blood concentrations to plasma concentrations, but also when not to convert blood concentrations to plasma concentrations. For instance, if you have a big variability between blood-plasma ratios for a certain analyte, you may wish not to convert a result, because you can have a very correct, you can have set up a procedure that allows you to derive a correct blood result, but actually then you can introduce a massive error by converting it to the plasma result. And actually you may wish not to convert it in several instances. In several other instances, it is possible to convert it and to get a really reliable plasma concentration that is based on an original blood concentration. I won't go into too much detail about this. The commutability of calibrators and QCs and real samples is very important. Actually, it comes down to the fact that you should be sure that your calibrators and QCs that are used in a certain procedure behave in the same way as real samples. And that's not always the case. And even if you participate to external quality control schemes, the samples that are sent around may actually be spiked samples, may not be real samples, and those may behave different from patient samples. And again, here I lined up, or I put in practice a very simple experiment. I won't go into too much detail, but it comes down to the fact that if you generate a spiked blood sample, and that should have the same result as an authentic sample, and you generate dry blood samples from that, you extract those, you should get the same result. Sounds overly simple, but we know that in many procedures, actually, that's not the case. Studies need to be set up in realistic conditions. If you wish to set up a study in Africa under tropical conditions, you must take into account, for instance, elevated temperature. You must take into account humidity, also during the sampling. So you must be aware of the context. Also, will it be supervised sampling, self-sampling at home? Transport conditions are important. So from that, also new insights can be gained. Just just one example. Again, unpublished work from my group, looking at tyrosine kinase inhibitors. Dasatinib was sampled via VAMS in the context of home sampling. So patients sampled at home at different points, different time points, and we look at trough concentrations. That's an advantage, and because if patients come to the hospital, they may have already taken their medication, or they may need to postpone taking their medications. Here we can really ask patients, okay, you can just in the comfort of your home environment sample right before you take your medication, and we will look at the variation in the concentrations. And you can see that in some individuals, for instance over here in the center, the different samplings give a very similar result. The gray shows the area in which you want the patients to be. And then, for instance, we can see that here patient number 4 is sometimes below the target area and sometimes above the target area. And so actually, if you don't have home sampling and you just have one sampling in the hospital, yeah, you can have this concentration or you can have that concentration. Actually, you don't know where the patient is. And here with this repeated sampling, you get insights actually into the behavior of the concentrations over time in a patient under realistic conditions at the home environment. And so patients are no longer a black box as they're being depicted over here. You really get insight into how a concentration of an analyte behaves over time in patients in their home environments. Because otherwise you could argue, what's the value of a clinical decision based on a single point measurement? In this patient, on one moment it's too high, on another other moment it's too low. And also if the patient presents to the hospital, coincidentally, that concentration that's measured at that time point could be too high or too low. And actually you can take a decision just based on one point without knowing actually the total context of the patient. Patient motivation is very important in order to get good samples. We've done several epidemiological studies, as I already said, and we feel that one of the key parameters to get good quality samples is that the patients really want to provide good quality samples. It's a patient-centric way. It's a convenient sampling, so sampling is possible at home. And in this context, also involvement of patient groups is important. We're now going to start up a new clinical study in which we will look at steroid hormones in children. And actually, there's a group of parents that's also being involved. And we really will ask those parents as well what they think about how the study should be conducted. Because if you don't have the patients or their parents in this case that are really motivated to also participate, then you know that you will fail. And in this context, it's important that there's a variety of devices available that are really also catering for the need of the patient. So to make sampling at home more accessible, more convenient for the patient. Just one example again from our own work using volumetric absorptive microsampling, in which we actually did this PET study, which I already referred to in 2019. And we showed that if we ask 3 successive samplings by non-trained individuals that didn't come to the lab, that only were shown leaflets and a movie, we had an overall success, so people that were successful in for the 3 samplings of 83%, roughly 92% per time point. Other studies have, and also including studies that we have performed, had lower success rates. But again, it all comes back to the motivation of the individuals involved. Here, for instance, the individuals were really interested in the behavior of their alcohol biomarker, and actually, the message that we gave them when they participated to the study was, you better get your sampling right, or you will have performed the sampling just for nothing because they weren't paid even to participate in the study. They just were interested in the behavior of the alcohol biomarker, and we said, if you don't sample right, we will not analyze the sample, and you will not get the results. And I truly believe that that was one of the key parameters, the key drivers for the success of this study. Actually, so this patient incentivization is very important, and also making the patients co-responsible. Actually, we're thinking of a situation in which you can really give patients insight into, for instance, the fluctuation of their concentrations, so that patients really know, yeah, perhaps it does matter when I take my medication. And if you can then move a patient rather from going all these fluctuating values to something like this, these blue values, then it can also be like a real positive incentive for the patient, like, okay, I changed my behavior, I changed the way I take my medication, and indeed, I see that my levels actually are stabilizing. Or you could also show them, like we did for the alcohol biomarker study, you could also show their value in the context of their peers, obviously anonymized. But then you could see, okay, all the other patients are doing well, and I'm fluctuating that much. Could be something related to the behavior of the patients, could also be something clinically relevant, and that also then gives insight into the medical doctor that monitors that patient. We also, in the context of the studies that we perform, typically ask about patient acceptance, and if we ask how painful people consider the finger stick, they typically don't consider it really painful. They typically consider it very easy, too easy, and very user-friendliness, very user-friendly. And if we ask them also if they would have a preference when compared to monthly sampling by a nurse or MD, the vast majority had a strong preference or moderate preference actually for the finger stick. So those were patients that actually went, or individuals that went through the process, knew what it was about, and like that could actually not just in theory answer the questionnaire, but actually they could answer. They were experienced by then and could really answer the questionnaire, knowing what finger prick sampling involved. Important is that microsampling should also be tailored to the need or possibilities of the lab. Clinical labs are typically focused on throughput, automation, and costs, and there's a challenge there still. Within microsampling, there's a possibility for increased throughput, increased safety, reduced hands-on time, and reduced hands-on time, apologies, and reduced risk on human mistakes. And different fully automated extraction systems are available or have been available because they have difficulty staying on the market because the field doesn't, let's say, the uptake isn't really taking place. So there's a variety of devices that have been on the market or in some labs are still present. We've demonstrated that it can work for several analytes. For instance, here, one example, anti-epileptic drugs, another example, immunosuppressants. And there's many other examples shown by other groups, also in the context of newborn screening, vitamin analysis, also in the context of COVID. Still, the thing isn't really happening. And although it is possible, we and others have shown that it is possible, it isn't really taking place. primarily because there's still some hesitancy and there's not enough demonstrations that it can work. Important is also that we should have realistic expectations. If we use dried blood microsampling, this is not the magic trick. This is not just some magic that can be done to make every study succeed. So we as lab persons are not the witches, are not the wizards that all of a sudden can make things work. For some clinical questions, microsampling may simply not be the best answer. And despite the improved stability of samples, for instance, dried samples, stability may simply not be good enough. I'll demonstrate that again with a sample from our own group, published last year, and it relates to the folate, so vitamin B9 analysis in dry blood microsamples. Actually, we initially aimed at monitoring women that want to get pregnant for their vitamin B9 status because it's really relevant in the context of prevention of neural tube defects. And microsampling would be a perfect approach to monitor those females. What we did was we, prior to conducting the entire study, we thought, okay, let's evaluate the stability under real-life conditions. So we sampled in the lab, put those samples in the post, and just send them again to the lab in order to have realistic conditions. When we did it in wintertime, everything was fine. The samples that we got back, the results from 5-methyl tetrahydrofolate, the main folate vitamin, were totally fine. However, in summertime, the results were off, and that actually led us to do an in-depth study on the stability of these folates. And then what we found out that if we send samples at elevated temperature, and it took a bit longer for those samples to reach the lab, for instance, at 37 degrees, then the results that we got were too low. So if you have controlled temperature conditions in which the samples can be sent, then results can be relied on. But if you don't have controlled temperature conditions, then it becomes a problem. And you should be certain that the samples reach the lab within just 1 or 2 days, ideally. So that's an important limitation. So it can work, but the conditions need to be right. And there's some things that you don't have fully under control, and obviously that complicates things. Intrinsically, there will always be some additional variation. The question is, what variation is acceptable for patient treatment and decision-making? So important in that context, maybe to talk to clinicians, for instance, and ask them, okay, what level of variation is acceptable? And important is also, how sure are we that we are still currently measuring the right thing? To accelerate the uptake of dry blood microsampling, two different approaches can actually be taken: a bottom-up and a top-down approach. I'm most in favor of a bottom-up approach, in which we actually show that it can work. Also show that patients are capable of self-sampling. Also show that it can provide reliable results, and show that there is an economic benefit. So like that, actually, that's really demonstrating that it is possible to really use it, and then try to convince others. An alternative is a top-down approach, and okay, it can also lead to the fact that there's more uptake. For instance, a policy change. If, for instance, dry blood microsampling is really included in some procedures, in some official procedures, or that's a bit more negative way of handling it, you could say, as if there's competition. Because, for instance, if one lab has a centralized facility to assess dry blood microsamples, it may take away samples from another lab, because samples can easily be sent via post, even internationally. And that may be a driver for that other lab also to embark upon the analysis of dry blood microsamples. So that's what I would call a top-down approach. And may also lead to an increased implementation and increased uptake. And in the end, I must say, I don't want to be negative, but because that could also be looked at in a positive way. In the end, it's all about the money. Cost evaluation of dry blood microsampling is really important. And this is just one example of a study from already quite a few years ago, and I believe that we need more of those studies. Typically, what's looked at if we talk about microsampling, that's solely the device cost. But if we talk about dry blood microsampling and compare it to conventional sampling, we should compare the overall thing. Because if in conventional sampling, we need someone who's medically trained. We need the patient that spends time to go to that medically trained person, or we need the nurse that needs to go home to that individual. So that also comes with a cost. But quite often those costs are different, or come from different pockets, you could say, and that is not always easy, because there's someone that needs to pay for the device, for instance, but that someone may not be the same and that benefits from the savings, from the overall savings. So there's still some challenges, some barriers to be overcome in that context. Overall, if we look at microsampling, I was already referring to black box, can lead to better use of medicines, and there's more and more expensive medicines, and we don't want to go those medicines wasted in our patients. So we want improved compliance, reduced side effects, and improved outcome, and this can really be helped, and this can be really facilitated by dry blood microsampling. So with this, I come to my conclusions. Dry blood microsampling analysis has evolved a lot since the establishment of newborn screening now over 60 years ago. It offers a wide variety of advantages, allows you to do things that are otherwise not possible. I showed some examples during my presentation. And there are various issues that are associated with microsampling and analysis of microsamples. That's why also we try to set up guidelines on how to validate procedures, but also on how to convert procedures on how to convert results from blood, capillary blood, to plasma. So I refer to the ones that are set up in the context of the ITDMCT. The last few years, a wide variety of approaches has become available that allow to deal with issues and challenges, also the hematocrits. It is being used increasingly, but the increase is quite slow in the context of TDM and toxicology. Important steps have been taken and continue to be taken towards implementation in routine, and provided they set up robust procedures, routine implementation is possible. If I think about the context of my lab, we have an accredited procedure for two analytes in the context of toxicology, and we run thousands, we analyze thousands of samples in the context of, for instance, driving license regranting cases. Important is, and then I come back to what I said in the very beginning of the presentation, is that more real-life data are needed, as well as studies that demonstrate that dry blood microsampling can really work, and that it can also really show a clinical and financial benefits. And during this presentation, I've primarily focused on, for instance, the therapeutic drug monitoring, but actually the same also holds true for clinical trials. And I was very happy that earlier this year, there was a publication in APS journal that came out, in which a couple of companies actually showed that patient-centric sampling can also really work in the context of clinical studies. And the one introducing me here, Niels Spooner, was also involved in there. So I was really happy to see studies like this, because we really need studies like this in order to move the field forward. And with this, I've come at the end of my presentation. I wish to thank my group, because I'm actually only just presenting the work that has been done by the people that actually do the analysis, do the work in my lab, shown over here in the picture. And I wish to thank you for your attention. And I'm open for questions, obviously. Thank you.
Thank you, Christophe. Very, very interesting. And with the Christmas hats, they're getting us ready early for the Christmas season.
Yes.
There's a few questions coming in. One from Chiara. Regarding blood to plasma ratio, do you recommend to calculate it and use it for each drug analyzed in every study?
There's a 21-page paper on that. Short answer is no. I would not recommend it to convert it in each study. And actually, the paper, which at this point is still behind the paywall, it will become open access. But anyone interested in getting it can send me an email. I can easily provide it. Actually, the paper really provides real life examples on how to do the conversion, when to do the conversion, when not to do the conversion. Just some examples. If there's a big variation in the blood plasma ratio within an individual or between individuals, it may not be possible to reliably convert blood concentration to plasma concentration. In other instances, you may have a time-dependent effect, so that the blood-plasma ratio differs depending on the timing of the sampling, and it is possible to take also that aspect into account into the formula that you set up to reliably convert blood to plasma concentrations. Actually, today a paper came out in APS journal in which we demonstrated that this can be done for paracetamol. And paracetamol and its metabolites. So the paper is just before this webinar. I saw that the paper was online.
Oh, cool.
And in other instances, you just need to know the hematocrits and can use the hematocrits in order to apply that in a formula to get a plasma concentration from a blood concentration. We recently demonstrated that for vitamin D, for instance, as an analyte. So it all depends on the context, on the analytes, but there's different ways in how to approach this.
Thank you, Christophe, for a comprehensive answer. It'd be great if that other publication, we've shared the guideline in the patient-centric sampling interest group, LinkedIn group, but it'd be great if you could share that other publication as well in the LinkedIn group, so that other people can see that. That'd be great. Next question was from Christian. Can you comment on the potential stability of DNA in DBS versus liquid samples collected by microsampling devices?
Yes, so like for DNA, actually, that's it's quite good. Stability of DNA is quite good. So there are studies that have looked at DNA that in dry blood microsamples that were many years old. And actually now, via conversations with a group at Ghent University, there's now also a group at Ghent University Hospital that is even looking at RNA, mRNA, for transcriptomics in dry blood microsamples. Because typically mRNA is considered to be really unstable as compared to DNA, but it works even for transcriptomic approaches via pre-soaking actually the dry blood microsampling device with a stabilizing solution so that you get the stabilization of the mRNA. So for the DNA, it works well. We've done that approach, but you never published it for genotyping, for instance, to compare genotyping with a phenotyping approach. And like I said, it works even for analytes which are renowned to be unstable, like mRNA.
I believe there are some commercial products out there for stabilizing oligonucleotides as dried blood spots. So, there are things out there. Next question, back to Chiara again. Do you give a quality score at the microsampling that the patients performed at home?
What was the question? I didn't get the first part of the question.
Do you give a quality score for the microsampling samples that are collected at home?
Yes. There's typically if we get samples in that are collected at home, we always score the quality, and that can be done in two ways actually. So we have a visual quality assessment. For instance, is the blood if it would be a blood spot, for instance, is it a blood spot rather than a smear? We look at both sides of the card, whether the diameter is big enough. But we also have a scanning system, and we can really look at the diameter of the spot that has been generated, and so we can also have like an automated good, not good assessments, and depending on the size of the punch that we want to make. For instance, for vitamin D, we make a 6 millimeter punch, but we know that the front of the card should, and that's in that context, the diameter of the spot should actually 7.5 millimeter, to be sure that for any hematocrits, we can actually reliably determine the concentration. If we consider other devices, for instance, the volumetric absorptive microsampling devices, the Mitra tips, there we also look whether they are fully filled, whether there's no whites left on the tips, and whether they're not overfilled, because overfilling can also occur if, rather than wicking up the blood, you have been actually depositing the blood on the filter tip, and like that you can also have oversampling. So those are things that we typically assess also in our lab. And if there's if the sample is not good, we don't analyze it.
No, thank you.
And important in that aspect is patient instruction, or the instruction of the individuals, or the instruction of the ones that help with the sampling, for instance.
Thank you. And I guess it helps that you a lot of these technologies, you have a replicate sample as well. So
yes, we always ask replicates. And for instance, if we would have a doubt about the quality of something, we can always analyze the replicates and then see if we would get the same result, yes or no. But for some samples, it's quite obvious that the quality may not be good enough. If you see a white spot on a Mitra tip, you know it's not completely filled. If you see really a very asymmetrical smear of dry blood, you also know already on beforehand that it's not good.
We're nearly out of time, but a quick last couple of questions. Teresa here says, how do you adjust for hot temperature mitigations? I'm not quite sure what that means, but I'm assuming, can you adjust for different temperatures?
We typically look at also temperature stability. Actually, what we wrote in the ITMCT guideline for the validation of the methods is what I would refer to as an accelerated stability or accelerated study to evaluate the extractability. And what we typically do is we compare the results of samples that have been put in a dry oven at 50 degrees for 2 days with fresh dry blood spots, and we want to get the same results. So that's irrespective of stability, because that obviously requires that a sample is that an analyte is stable, and that's extractability. And the reason why we look at this elevated temperature is because it's a bit inconvenient if you have to wait every time 2 weeks for a stability experiment, and whether your extraction really is capable of extracting the analyte reliably from a very thoroughly dried spot. That's why we came up with this accelerated elevated temperature experiment.
Yeah, I've always loved that experiment, Christophe. I think it's a little bit of genius. Last question from Maros. Can you comment on stabilizing vitamins in dried blood? I'm not quite sure which vitamin it is. Obviously, vitamins vary quite a lot.
Yes, and there's several approaches that can be applied. For instance, you can have pre-soaking of the filter paper with, for instance, ascorbic acids or with dithiothiotol. So there's several approaches that can be tried. For instance, for the folates, we tried several approaches to come up to try to stabilize the folates, but we failed. And nothing was good enough in order to provide sufficient protection for the folates. But I know for some vitamins it may work, and providing or pre-soaking the device, whether it's a microsampling device or filter paper with an antioxidant, can actually provide protection and can lead to stabilization of the vitamins.
Thank you, and Maros said thank you as well. Okay, thank you very much, Christophe. I think that's all the time we have. I'd just like to finish off before we leave everyone, just reminding everyone, if they're not members of the patient-centric sampling interest group, you can join us for free through our LinkedIn group using the code on the right-hand side there. There's a lot of information on our website. We're always welcoming extra people to get involved with our group and help us with our mission to make enabling patients to take control of sampling. We've got another webinar coming up in a few days, just a couple of weeks. Stephen Pennington is going to be talking to us about towards personalized medicine, blood microsampling and mass spectrometry-based proteomics. And then our final webinar for the year in December is Carissa Jones, Bill Ains, and Gerald Finken talking to us about expanding zip codes, a novel approach to reaching and engaging clinical trial participants across the United States. So, very exciting, a variety. We've got a lot of webinars lined up for you. We've now got webinars booked up until January 2027. So a lot of content to come, and obviously a lot of historical content on our YouTube site and on our website. Just like to thank our sponsors again who make all of this possible. Sorry, that slide is not changing. There we go. All our partners and sponsors who make all of this possible. And obviously, Christophe, thank you so much for giving us yet another wonderful webinar, giving us a little bit of history, a little bit of what the challenges are, and a few thoughts about where we might be going and how we need to get there. Great insights as ever, Christophe. It's an absolute pleasure to be able to host you in one of our webinars, and we look forward to talking to you again maybe in another couple of years' time.
Okay, thank you for having me.
And thank you for all of you that attended today. This webinar will be available in a few days on our YouTube channel for free. And please keep registering for our webinars and supporting PCSIG. Thank you all.